All right, the second part of this lecture concerns itself with the requirements that microbes have in order to grow well. So we're going to talk about growth requirements that are physical in nature, and then we'll talk about growth requirements that are chemical in nature. And all of those things are meant to determine for us the optimal environments in which these microorganisms grow so that they reach their biotic potential. So what physical and chemical things can we change or manipulate in order to get these microorganisms to grow optimally? First up, we're going to talk about life in the presence of oxygen, which is kind of interesting to find out is actually fairly difficult to accomplish because oxygen will naturally convert to what are called reactive oxygen species. So reactive oxygen species, which you'll sometimes hear called toxic oxygen species or free radicals, are chemically reactive oxygen species. So molecular oxygen, O2, is very stable, but the problem is that oxygen molecule, as it's being manipulated in the cell, can be converted to these reactive oxygen species or free radicals, and they're incredibly damaging to the cell. They convert to superoxides or hydrogen peroxides, and those things happen as a result of aerobic respiration. And once those superoxides and hydrogen peroxides are produced, they're kind of, if you picture like the Tasmanian devil, they just spin up and they zoom all over the cytoplasm of the cell, and as they bang into DNA or RNA or fatty acids or amino acids or enzymes or anything else in the cell, they just bang into them and break them. And if they break enough of them or cause enough damage to the DNA, it kills the cell. So if you are going to live in the presence of oxygen, you have to have enzymes or some ability to deal with these reactive oxygen species. You might remember me mentioning reactive oxygen species when we were talking about microbial cells. I mentioned that there are magnetosomes in which cells use iron oxide to destroy hydrogen peroxide. So that's one option that you have if you're living in an aerobic environment. But typically, we think of two enzymes being used in order for cells to be able to grow in the presence of oxygen, and those are superoxide dismutase and catalase. And these are considered antioxidants because they undo the oxidation of that reactive oxygen species and convert it back to its reduced non-reactive form. So if you take this superoxide anion and you expose it to a superoxide dismutase, the superoxide dismutase will convert that superoxide anion to hydrogen peroxide. So now you don't have that superoxide anymore. The problem is you have hydrogen peroxide. So most of the time, superoxide dismutase is found in organisms which also have a second enzyme, catalase, which converts hydrogen peroxide to the very stable and non-reactive water molecule. So calling this an antioxidant might help you to connect it to the real world, right? People talk about free radicals and talk about taking foods that are rich in antioxidants in order to help them to deal with free radicals that can contribute to disease or even aging, right? So wrinkles are thought to be a result of free radical damage to skin cells. So if you take an antioxidant lotion or topical ointment, you can assumably undo some of that damage. But what we're talking about is the damage that causes death to cells, not single cells of a multicellular organism, but the cell of a unicellular organism, which causes it to die. one of these two, superoxide dismutase, we don't test for in lab, but we do test for catalase because it's a really easy test to do. All you have to do is take a colony of cells and add hydrogen peroxide to that colony, and if the colony bubbles, you know that it produces catalase. If the colony does not bubble, the colony is a catalase negative organism. So in this photograph, you have on the left-hand side a catalase positive organism, and on the right-hand side a catalase negative organism. In the next slide, we're going to talk about how the presence of catalase and superoxide dismutase allow organisms to live in and thrive in aerobic environments. The first of those is oxygen, and we've talked a little bit about oxygen in the past. You've seen some stuff in lab about the growth requirements of organisms in terms of their oxygen requirements, and you know that one of the media that we use in order to determine the oxygen requirements for a given microorganism is something called fluid thioglycolate media. And fluid thioglycolate media is described in detail in lab exercise 11. What I'd like to do here is not so much describe the medium, but describe what happens and what that can tell us about microorganisms. So FTM has a reducing agent in it, which takes the oxygen that diffuses into the medium from the environment and reduces it to water, creating an anaerobic environment throughout about the bottom, say 70 to 80% of the tube. So when we inoculate bacteria into this medium, they will attach themselves to tiny pieces of agar and they won't move vertically through the media. So where you put them, they will either grow and make the medium turbid there, or they will die because whatever oxygen level is there is not appropriate for them. And so where they grow in this media can tell you what their oxygen requirement is. So in group A, you have organisms who will only grow at atmospheric concentrations of oxygen, and we call those organisms obligate aerobes or strict aerobes. Strict aerobes, like you and I and like other organisms like Bacillus subtilis, will only grow in the presence of oxygen. However, they have to have special enzymes in order to prevent the reactive oxygen species that are produced as a byproduct of aerobic metabolism to protect themselves. So they produce both catalase and superoxide dismutase to protect themselves from the reactive oxygen species that are being created as they undergo aerobic respiration. The second group are the exact opposite of that. Those are obligate or strict anaerobes, and those guys will only grow in environments where oxygen is not present. And the reason for that is that these organisms lack those enzymes that would neutralize the reactive oxygen species. And so when oxygen is present, the oxygen converts to these reactive oxygen species, and then the organism dies in the presence of oxygen. So they're only capable of growing where there is no oxygen present. Next, we have two really interesting organisms. Notice both of their names have anaerobe in them. We have a facultative anaerobe and we have an aerotolerant anaerobe. So both of these organisms can grow anaerobically, as is evidenced by their growth in FTM. They also can grow aerobically. So they are not strict or obligated to anything. They can grow in both environments. The difference between the two of them can be seen in the word that precedes anaerobe. The facultative anaerobe is an organism that can grow anaerobically but does not prefer growing anaerobically. Whenever you see facultative, whatever follows it is not its optimum. It's just something it's capable of. So a facultative anaerobe can grow without oxygen, but it does not prefer that. So their growth pattern is to grow really well in the presence of oxygen and to grow not as well in the anaerobic environment. And that has nothing to do with these neutralizing enzymes. superoxide dismutase and catalase, it has more to do with the fact that when oxygen is present, they will undergo aerobic respiration, which, as you know from previous lectures, gives you the most ATP per pound. And so they can make way more ATP, they can grow way faster when oxygen is present. When oxygen is not present, they will either switch to fermentative metabolism and make very, very few ATP per molecule of glucose, or they will switch to anaerobic respiration, which, though it is better than fermentation, still does not produce the amount of ATP that it's produced during aerobic respiration. So they will grow anaerobically. They do not prefer it. They are facultative anaerobes. Next up, we have our other anaerobes that have a caveat to their name. These are the aerotolerant anaerobes. So that tells you what they're doing. Unlike the obligate or strict anaerobe, these guys can tolerate an aerobic environment. They tolerate oxygen, but they never use oxygen. So oxygen has no effect on their growth. It does not increase their growth like the facultative anaerobe, and it does not inhibit their growth like the obligate anaerobe. The aerotolerant anaerobe can grow in the presence of oxygen, but it does not utilize the oxygen, so it doesn't increase its growth. And these guys have superoxide dismutase to neutralize the reactive oxygen species, but they either are fermentative, like genus Streptococcus, or they just don't grow preferentially with oxygen because they lack catalase. Finally, we have the microaerophiles. And microaerophiles, they like oxygen, but they like just a little bit. So these guys grow in 5 to 15% oxygen. And if you increase the oxygen level above that, then the reactive oxygen species that are made will become lethal and the organisms die. So they cannot grow anaerobically and they can't grow in full atmospheric oxygen. So when we grow bacteria in FTM, it's super cool, right? Because in this one tube, we can tell not only can you grow aerobically or anaerobically, but do you use oxygen or do you just tolerate its presence? One other way that we can look for the ability to grow anaerobically is to use an anaerobic jar, which is pictured here on the left, which, like the fluid thioglycolate medium, has a reducing agent which converts all of the oxygen in that sealed container to water. And if you're working with a strict anaerobe, you can do your inoculations in an anaerobic hood. So rather than doing those inoculations on the lab bench, which would be exposed to oxygen, you can actually perform your inoculations in one of these anaerobic hoods. So one example, I know this is kind of disgusting, but one example of an anaerobic microorganism that's clinically important is that same Clostridium perfringens that I mentioned has a 6-minute generation time, because that is capable of growing in anaerobic tissues and can very quickly cause gas gangrene, and that's a very common issue for people who have diabetes. They have poor circulation into those tissues. The anaerobic tissues, because they have poor circulation, don't have a lot of immune system interaction. They also tend to have neuropathy, so they can't feel their feet. They don't know that they have that infection, and that infection can very, very quickly get out of control because of that very rapidly reproducing anaerobic microorganism. Next, we have pH. So organisms all have a fairly narrow pH zone in which they do well. And on either side of that very narrow zone, they very quickly don't grow at all. So if we take the middle of the pH scale, the pH 7, we have a neutral pH, and organisms who thrive at a neutral pH are called neutrophiles. Organisms who thrive at an acidic pH are called acidophiles, and organisms who thrive at a basic or alkaline pH are called alkalinophiles. Notice again on both sides, right, whether it's too acidic or too alkaline, they very quickly perish and are not able to grow at all. Now, regardless of the environment you're talking about, When the pH gets too low for the acidophile or the neutrophile or the alkalinophile, that causes the organism to tumble if they're motile. So they will all try to move out of that low pH environment and hopefully move into one that is more at their optimum. In addition, in all of these environments, the surface proteins of these microorganisms are incredibly stable, and they adapt very quickly to changes in the pH. In addition, the proteins that are found on the membrane or surface of the cells will create these really interesting microenvironments where they actually gather protons in order to neutralize an alkaline environment, or they grab hydroxyl ions from the environment in order to neutralize an acidic environment. They also buffer changes in pH using their nucleic acids, using the phosphate backbone of their nucleic acid. They buffer their environment, and they have basic or alkaline or acidic amino acid side chains, which allow them to adapt to environment. So in any environment, whether you're an acidophile, a neutrophile, or an alkalinophile, low pH makes you move out of the environment. Your surface is very stable to changes in pH and can change, right? It can change and gather protons to offset an alkaline environment or gather hydroxyls to offset an acidic environment. If they get inside the cell, the DNA and the RNA can serve to buffer those so it can lose protons in order to neutralize and bring down the pH, or it can gather protons in order to neutralize and bring up the pH, and then their amino acid side chains can gather protons and hydroxyls as well. And it's really crazy. So whereas our cells have a very narrow pH within which they can operate, right? Typically we think of the pH for human cells to be 7.4 or thereabouts, right? 7.35 to 7.45 or something like that. E. coli, which is a neutrophile, you can put that cell in a pH range from pH 4.5 to 7.9. So let's just say 5 to 8. And the inside of that E. coli cell, the cytoplasm, will only register a 0.1 unit change in pH. So you have an environment, right? Remember pH scale, each number is a 10-fold difference. So when you say from 5 to 8, 5 to 6, so 6 is 10 times more alkaline than 5, 7 is 100 times more alkaline than 5, 8 is 1,000 times more alkaline than 5, pH 8 is 1,000 times more alkaline than pH 5, and the inside of that cell will only change 0.1 units. So you have a 3-unit change and a 0.1 unit change. So they're incredibly adaptable and able to withstand changes in pH, much more so than human cells or animal cells. And that allows them to inhabit these environments that change very, very quickly and routinely and adapt to those changes. Now, when you talk about cells who live in an alkaline environment, those that are alkalinophiles, they typically also live in salt-rich environments, and they utilize sodium and proton antiporters. So they utilize this active transport, much like the sodium-potassium pump you talk about in general biology, in order to move sodium along its concentration gradient and then pump protons opposite their concentration gradient. So they have these voltage-gated channels where sodium moves in and then sodium is pushed out as hydrogen ions are brought in. So that's how they work in alkaline environments. In acidic environments, those cells who are acidophilic tend to have membranes that are not very permeable to protons at all, but they also will utilize that natural proton gradient in order to undergo chemiosmosis. So they don't even have to use the electron transport chain. All they have to do is allow those protons to flow down their proton gradient from the outside inside, and then they just collect up those protons and expel them in vesicles in order to get the protons back out of the cell. So take home, all cells are capable of living in a much, much wider range of pH than animal cells or fungal cells or plant cells. And that's because they can move out of that low pH environment. They have very pH stable proteins and amino acids. Their membranes will create a microenvironment around the cell that is more neutral than the surrounding environment. And if the protons or hydroxyl ions get into the cell, the DNA chromosome, the phosphate backbone of the chromosome can be used to buffer and keep the internal pH very stable, even though the external pH might vary wildly. So one example of acidophilic microorganisms is one who also is a thermophilic organism. So in Yellowstone Park, in the geysers, we have these huge pools where thermoacidophilic microorganisms both survive, thrive, and require hot environments and acidic environments. In addition, there are haloalkalophilic microorganisms that like to grow in both salt, rich, and alkaline environments. So one example of acidophilic microorganisms is one who also is a thermophilic organism. So in Yellowstone Park, in the geysers, we have these huge pools where thermoacidophilic microorganisms both survive, thrive, and require hot environments and acidic environments. In addition, there are haloalkalophilic microorganisms that like to grow in both salt, rich, and alkaline environments. Next up, we have temperature. And the reason that temperature is important to microorganisms is that temperature has an effect on enzymes, and that's something that we talked about in the metabolism lecture a little bit. At optimal temperature, enzymatic reactions are occurring at a maximal rate. Anything above that optimum is in danger of denaturing the proteins and perhaps lysing the cell due to heat. Anything below that slows the interaction between substrate and enzyme, and because substrates and enzymes are not encountering one another, ATP is not being produced and the cell is growing very slowly. Now, one thing to note that's very important, notice I said at higher than optimal temperatures, the cell dies. Proteins denature, they don't come back after they denature, and that will kill the cell. If the cell gets hot enough that it undergoes thermal lysis, obviously the cell doesn't come back from being blown up. So high temperature, higher than optimal temperature, kills microorganisms. Now notice on the low end, minimum growth does not become minimum because of death. It becomes a minimal temperature at which the organism will grow, because at that point, the membrane becomes so solidified that nutrients can't be transported into the cell, and enzyme reactions are going so slowly that growth does not occur. So minimum temperature does not kill the cell. So low temperature can control the growth of a cell by slowing growth, but it does not kill cells, whereas maximal temperature exists where the cell actually dies. So minimum, slow, lower than minimum, very, 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 very slow growth. As temperature increases, enzymatic reactions occur at increasingly rapid rates and growth occurs faster. When you get to the optimum, everything's happening optimally. And if you get hotter than that, enzymes denature and the cell dies. So notice there are no degrees on the x-axis. It's simply temperature, and that is because the optimum for organisms varies widely when we talk about microorganisms. Those organisms who grow optimally at body temperature, right in the middle of this range, are called mesophiles, right? They're the middle likers, right? Middle. Meso means middle, so a mesophile likes it in the middle. They like it right at body temperature. So at higher than body temperature, enzymes start to denature. At lower than body temperature, everything's going slower, slower, slower. Those organisms who grow, thrive, and inhabit higher than body temperature environments are called thermophiles. And thermophiles, typically we talk about growing optimally at 60 degrees Celsius or higher. Hyperthermophiles grow optimally at or above the boiling temperature of water. And then on the low end of the scale, psychrophiles grow at between 4 and 10 degrees Celsius optimally. But and psychrotrophs are a really interesting group of organisms. So psychrotroph, you could kind of compare to the facultative anaerobe, right? Whereas the facultative anaerobe can survive anaerobically but doesn't prefer it. The psychrotroph can grow in cold, but it doesn't prefer it. So psychrotrophs are those organisms which can grow in cool temperatures but don't prefer it. And really, for this one, the psychrotroph, the problem with a psychrotroph is his optimal temperature for growth is usually higher than what is indicated on this chart here from the Tortora textbook. Psychrotrophs typically grow optimally at mesophilic temperatures, but can grow at low temperatures. And the problem for us with psychrotrophs from a human perspective is that they can cause food spoilage because they're able to grow relatively well at refrigerator temperatures. So a mesophile doesn't really grow at refrigerator temperatures, whereas a psychrotroph can grow at refrigerator temperatures and cause food spoilage. Before we move on to our last physical growth requirement, just a word about food spoilage and trying to get your food to a proper temperature in order to prevent food spoilage. And we're going to do that by looking at two different experimental setups. The first in blue is some rice that's just been made. So if you've ever made rice, you know you have to keep it at a high temperature. It's very hot. You keep it there for long enough, right? It boils, and then you have your rice. And so you have this steaming hot rice, and you only eat a little bit of it, and you want to save the rest of it. So the question is, what do I do with this rice? I'm going to put it in the refrigerator in order to save it. So do I put it in a shallow container, or do I put it in a deep container? Do I transfer it to a Tupperware container that is shallow and larger, or do I keep it in the container that I served it in? And the problem with the depth of containers has to do with the surface area of the containers. This blue container has a very large surface area to volume ratio, and so it will cool differently than the red container. And if we look at the temperature of the inside of these containers, when we put them in the refrigerator, notice that the blue container, whose temperature is shown in blue, very quickly goes from 70 degrees down to refrigerator temperature, right? It does that in about 4 hours. If we compare that to the red container, it goes from 80 degrees or 70 degrees, like somewhere up there, really hot. And notice even after 8 hours, it's still warm in the refrigerator. So now we want to apply what we know about psychrotrophs to this concept, and even mesophiles to this concept, right? Mesophiles and psychrotrophs grow optimally at 37 degrees Celsius, and they grow well at between 15 and 45 degrees Celsius. So as you put these foods into the refrigerator, initially they'll be too hot for a mesophile, which could also grow in your body, to grow. The blue container will move into that area of temperature wherein a mesophile will grow well fairly quickly. And so those microbes that are present in this food will start to undergo binary fission. Now, they will undergo binary fission until that rice hits 15 degrees. And that takes about an hour. So within that hour, each of those cells could undergo, let's say, 3 rounds of replication, which is not too bad, right? Compare that to the red container. The red container hits what is called in the food service industry the danger zone at about 3 hours. And notice even at 8 hours, so for 5 hours, this thing is in the danger zone. Any organism that's in there is reproducing. And remember, if it's 3 rounds of replication an hour, then by the time you get to 7, you've got more than 1 million. And that, my friends, is how you get food poisoning when you take food off the stove and stick it into the refrigerator. Because if you don't transfer it into a shallow container, then the food takes so long to cool past the place where mesophiles can grow that you end up having an abundance of microorganisms in the food when you take it out of the refrigerator. So that's a definite consideration for you as you're trying to keep food safe in your homes. And just to talk about thermophiles, so thermophiles a lot of times are also barophiles. They require high pressure, and a lot of them exist in the deep sea hydrothermal vents. This is a barothermophilic community in the North Pacific Ocean off Vancouver Island in the Juan de Fuca Ridge. So again, kind of interesting, we have these polyextremophiles. We had haloalkylophilic organisms, thermoacidophilic, and now we have a barothermophilic microorganisms existing at high temperature and high pressure. All right, the last thing that we're going to look at for physical growth requirements is salt. And the reason we look at salt is because salt can cause osmotic or turgor pressure based on the relative concentration of salt in the environment and in the cell. When the concentration of salt outside is lower than inside, then water wants to rush into the cell, and that can cause the cell to lyse, right? Which is one of the reasons why bacteria like to have cell walls, because cell walls resist that turgor pressure, right? So I made turgor big there, so you think, oh, turgor pressure causes the cell to get big. Now, if the concentration of salts and solutes inside the cell is lower than the outside of the cell, then that can cause things to leave and the cell will shrink under what's called osmotic pressure. So osmotic pressure causes the cell to shrink. Turgor pressure would cause the cell to get larger. And the real problem for most organisms is that they encounter osmotic stress. They live in an environment where they have a high concentration of things inside their cells and water wants to rush in, right? So how do they deal with that? They have a cell wall which resists that turgor pressure. But you can live in an environment where you have more things outside, like a high salt environment like the ocean or the Dead Sea or the Great Salt Lake or Mono Lake or something like that. And in that environment, you exist in a state where water constantly wants to leave the cell, right? Osmotic pressure makes the water want to go outside and dilute what's out there. And so you have to come up with a way of keeping the water inside of the cell. So basically you have to come up with a way of having enough stuff inside that the water is balanced. So in order to do that, cells will make what are called compatible solutes. And compatible solutes are dissolved in water. That's why, right? Solutes are dissolved in a solution, so they're dissolved in the cytoplasm of the cell, and they are compatible both with the life of the cell and compatible with the salt outside. So what a compatible solute will do is sit inside as an inert molecule and make the water want to stay. And so they increase the halotolerance of the cell, and they have to have a couple of traits in order to make them work. Number one, they have to have a high solubility in water. So you have to be able to have a lot of these compatible solutes inside the cell that can be dissolved in water. They also need to have a high permeability so that if the osmotic stress changes, they can go outside of the cell. They also, very importantly, unlike salt, need to be metabolically inert so that they don't damage any of the portions of the cell. So they increase the halotolerance of the cell by making water want to stay inside the cell, and they have to have a high solubility, high permeability, and they need to be metabolically inert. And there are several different kinds of compatible solutes, and we're really not going to function on them. What you want to know is just how they work, right? So they increase the halotolerance, and they do that because they have a high solubility, right? They'll dissolve in the water, they'll hold the water into the cell, they don't do anything inside the cell that damages any of the chemical reactions, and then when the cell doesn't need them, they can be released into the environment very readily. So we don't typically talk about low salt environments, we talk about high salt environments. And we compare those to the salt that our cells like. So if we are non-halophilic, we consider non-halophiles to like a salt concentration of 0.09%, and anything that likes it higher than that, we give a halo prefix to. So halophiles like it to be saltier than 0.9, and typically they want it to be at least 3% salt. So that's marine organisms. Marine organisms, the ocean is 3% salt. Halotolerant organisms do not prefer a high salt environment, but like the aerotolerant anaerobe can tolerate a high salt environment. So they grow best in a non-halophilic environment, but they can grow in fairly high concentrations of salt. Extreme halophiles grow, thrive, require concentrations of salt that get toward the saturation point of salt in water, right? So they can grow at 30% salt, and it doesn't get any higher than that. If the salt concentration gets any higher than 30%, the salt comes out of solution as crystals. And so halophiles like it at least 3% salt. Extreme halophiles like it at 30%. Non-halophiles like us like it at 0.9%, and halotolerant organisms can survive at high salt but don't prefer high salt. And as I mentioned, one of the organisms that likes it to be salty also likes it to be alkaline, and that is our haloalkalophilic bacteria, which can be found in environments like Mono Lake, which has a salt concentration about 3 times that of the ocean and has a pH of about 9.5. So here's one other example of haloalkalophilic microorganisms that's shown in the OpenStax textbook, Lake Natron in Tanzania. And that red color that you see is actually a pigment produced by those haloalkalophilic microorganisms, and that pigment actually helps them to survive in the presence of high salt. Okay, that, my friends, is all of the physical growth requirements. We had temperature, oxygen, pH, and salt as our physical requirements for growth. We're going to be examining 3 of those through lab activities. We'll be looking at oxygen, temperature, and pH. Just because of the difficulty And now we're going to switch our attention to chemical growth requirements. Cells typically have 6 chemical growth requirements. They require carbon, they require they require nitrogen, they require sulfur, and they require phosphorus. Carbon, of course, is the backbone of all of the biological macromolecules, proteins, polysaccharides, nucleic acids, and lipids. Nitrogen is found in nucleic acids and proteins. Sulfur is found in proteins. Phosphorus is found in nucleic acids. And so all cells require some form of carbon, nitrogen, sulfur, and phosphorus. Cells, just like your cells, also require vitamins and minerals. Trace elements are the vitamins that are required by a particular microorganism, and organic growth factors are the minerals that are required by these organisms. So as you can see here, they require proteins both for their flagella, their membrane proteins, their channel proteins, pumps, and all that sort of stuff, receptor proteins. The cell wall has a protein component. There are proteins in the cytoplasm. Nucleic acids, of course, the DNA is found as part of the chromosome and the plasmids. RNA is found in the ribosomes, the messenger RNA and the tRNA. Polysaccharides are found in the glycan portion of the cell wall. They're found in the lipopolysaccharide outer membrane, and of course polysaccharides are storage granules are filled with polysaccharides as an energy source. Lipids are part of the lipopolysaccharide outer membrane. They're part of the phospholipid bilayer membrane, and of course fats are used as a storage molecule as well. So you have polysaccharide granules and lipid inclusions and all that fun stuff in order to store energy for long-term use. So when we're growing microorganisms, we work really hard to figure out what kind of carbon do they like, what kind of nitrogen do they like, what kind of sulfur, what kind of phosphorus, what vitamins do they need, what minerals do they need in order to grow well, because most of the time in the microbiology lab, we're trying to get our microorganisms to grow well so that we can learn about them. So I want to take just a moment to show you a couple of experiments and how very small changes in the chemicals that we provide to microorganisms can cause very drastic changes in their rate of growth. So first off, we're looking at a microorganism who's grown in a medium whose nitrogen source is inorganic. The nitrogen source that's being fed to this organism is ammonium chloride. And when we do that, the generation time of this organism, not 6 minutes or 20 minutes, but 85 hours, which means that you have to wait a very long time to learn anything about this organism. So you start tweaking the recipe and trying to decide whether or not you can figure out a better way of feeding this microorganism. And one of the things that you can change is the ammonia. You can say, okay, instead of giving him an inorganic form of nitrogen, I'm going to try an organic form of nitrogen. So you give him glutamine, which is an amino acid, and lo and behold, the generation time drops from 85 to 30 hours. Now, that's still not 6 minutes or 20 minutes, but it's about a third of what the generation time was previously. So sometimes making minute changes in the media makes for drastic changes in the generation time and the biotic potential of the organism that you're growing. Now, for us, that might mean differences between these two media here. For instance, on the left, you have nutrient agar, which is a complex medium for the growth of heterotrophic bacteria. So it's complex, which we talked about earlier on in lab, which means we don't really know exactly what's in it, but it works pretty well for most heterotrophs. It's got peptone in it, it's got beef extract, and it's got some salt. Now, if we want to be able to grow Escherichia coli maximally, we might switch from nutrient agar, just this complex medium, to something that's been chemically defined, where now we know exactly how much of every single thing is inside this medium. And when we do that, we can grow E. coli faster. The question you want to ask yourself is, does it really matter whether the generation time is 20 minutes or 30 minutes? Is that really going to change things? And is that change great enough that I want to have a chemically defined medium for every single microorganism that I want to grow in my lab? And for the most part, in a general lab, no, the answer is no. You don't need to get the generation time down to 20. 30 is just fine, because 30 minutes will still provide you with ample growth of the microorganism in a 24-hour period. So most of the time, we don't work with chemically defined media for microorganisms. We work with complex media, because complex media like nutrient agar or Luria bertani agar or tryptic soy agar or things like that will grow heterotrophs very nearly optimally, and that's good enough for us. When we're talking about something like the isolate I just showed you data for, that can go from 85 hours to 30 hours, now you're talking about a change that's significant. And I want to show you, not because I want you to memorize any of these details, but I want to show you just how small a change that really is. This medium here is all of the things that go into a medium for growing purple sulfur bacteria photoautotrophically. One of those ingredients is nitrogen. They need a nitrogen source. In the first example, which yielded an 85-hour generation time, we used 0.5 grams per liter of ammonium chloride, which we switched to glutamine for the second experiment and reduced the generation time to a third. So even small media changes really do matter. And if you're talking about something that has a long generation time and does not have very high biotic potential, then this kind of a change might be something that you want to investigate. But for us, we normally just grow hemoheterotrophs in a general complex medium like nutrient broth or tryptic soy broth or something like that.